So I'm very happy to introduce you to today's speaker, which is Adrian Egli. Adrian Egli is a professor at the University Hospital in Basel, and he is the lead of the personalized sepsis study driver project. It's a driver project which is really a real driver for SPHN. So his project is really testing and developing a lot of things which really help us for the general infrastructure. The project itself is about finding biomarkers to detect sepsis and also to predict the outcome of a sepsis. With that, I give the floor to Adrian Egli. I'm looking forward to hear your presentation.
Okay, thank you very much for the very nice introduction. So this is actually my first lecture I give via Zoom, so please excuse if there are some technical difficulties, so I'll try my best. So first of all, the study has, or this talk has the longest title ever, I realize. So shortly, I will present our attempts to build a sepsis network, the personalized Swiss sepsis study, during the next about 45 minutes, and then there's plenty of room for discussions. So this is obviously a project which is financed by SPHN and PHRT, and so I'm very thankful for that, and I will also introduce you right away. I have to check, there's an issue with sharing. So what I will do is during these next 45 minutes, I will introduce you to our team, because I'm basically just selling the car to you, but obviously there are lots of people involved in this project. I will talk about why do we focus on sepsis and why I really think this is an excellent disease, let's say, for a driver project because of the heterogeneity. I will talk a bit about current diagnostic bottlenecks and what is the potential of digital biomarkers. Then I will certainly also dig into the details of the framework and the setup we have, our current achievements, which are very nice, and I'm pleased to see how this project makes now very nice advances, and then I will sum up and draw some conclusions, also maybe from a more general perspective for SPHN as a whole. So let me really start with the team. Without a fantastic team, it wouldn't work, and I couldn't list here all people involved because there's a lot of technical people involved at every university hospital, and they are actually doing the main work at the moment to build up the framework. I just show you basically, let's say, the academic interested groups. All university hospitals are included. In some cities, also the universities themselves, then ETH Zurich and SIB. And where I'm really, happy to say that in each city, we always have the intensive care unit, the infectious diseases, and the clinical microbiology division involved in this endeavor. And also, obviously, lots of ETH associated groups who will help us analyze all the data we're going to collect and produce. So why do we have a focus on sepsis? So first of all, I think we have to understand a bit what is sepsis and why this is an important need in the medical field. So first of all, sepsis is a very heterogeneous disease and has a profound impact. The course and outcome of sepsis depends on different aspects. So first of all, there's the causative pathogen, then the infected patient, obviously. So if we think about ourselves, we're all genetically diverse, we have different gender, age groups, etc. are affected. And this all influences, obviously, the outcome during an infection. And then what is often forgotten, but what is also very, very important, especially in sepsis, is the timing. So at what stage does a patient present him or herself to an emergency ward? If this is already progressed, then obviously the outcome is much worse. And this is very nicely summarized on the right graph, which you can see here. On the y-axis, you can see the fraction of total patients from 0 to 100%. On the x-axis, you have time of hypertension onset, and hypertension here reflects the septic shock. So this patient is really severely ill. And what you can see here, the black bars, they are rapidly going down, and this is the survival fraction. So we already start at 80%. So, you know, sepsis is a severe disease, obviously, right from the start, but then it rapidly declines. I mean, if you check, for example, 5 hours after hypertension onset, the survival rate is about 50%. And what is obviously very important is then to initiate antimicrobial treatments. And this is indicated by the gray bars. So rapid diagnostic of sepsis and the identification of the bacterial agent are very critical steps for a successful treatment, because if you know who the enemy is, then you also know how to potentially fight the enemy. So there are international well-accepted definitions for sepsis, and there are 2 definitions. The first one, sepsis 2, I learned in med school, so it's quite aged, I would say, and the newer definition is now the new standard, sepsis 3 definition. Sepsis 2, basically, just to quickly repeat that, this is a proven or suspected infection. So infection is really an important aspect. And then you have SIRS, and SIRS stands for Systemic Inflammatory Response Syndrome. And this describes basically the inflammatory response of the human body. So elevated body temperature, heart rate, and there are a couple of other factors included here. Now, if I walk from the basement to the 2nd floor, then I would also have a SIRS. So my body temperature would rise, my heart rate would certainly rise above 90. And so obviously this is a definition which is very, very sensitive. So we will include a lot of people who have a sepsis, because this criteria is easily fulfilled. The second definition, and there they try to adapt that, still has the infection aspect. But then there are other factors which are included, the so-called SOFA score or SOFA factors. And this stands for sepsis-related organ failure assessment. And this is, let's say, factors which are describing the mortality of the patient. So you have in the first row, when you look at partial oxygen ratios, these are lung functions. Then you have neurological scores like the Glasgow Coma Scale. You have heart outputs with the mean arterial pressure. You have liver functions with the bilirubin, serum creatinine, which gives you information on the kidney function, and so on. So this really focuses more on organ failure. And until you have organ failure, this is a bit later progressed in sepsis, and therefore this definition here is less sensitive but far more specific. So if somebody fulfills these criteria, you can be pretty sure that that person has a sepsis. So maybe the truth is somewhere in between, and this is also interesting to actually explore new markers which may help you to define sepsis. So just to quickly sum it up, you have a diagnostic element, the infection. So you need to know what the pathogen is, or you need to suspect the pathogen. But then at the same time, sepsis 2 focuses on the inflammation, and sepsis 3 is basically an assessment for the risk of mortality. Now, other people have thought about how we could improve this definition, the clinical phenotypes, which are very diverse in sepsis. So you can imagine a 20-year-old who has a urinary tract infection and then develops a urosepsis is very different from a person who is 80 in an elderly home and has an influenza and then develops a pneumonia, and from that a sepsis. So this is really, really diverse. And there's a fantastic paper by Chris Seymour published in JAMA last year. And Chris was actually a guest here at the SPHN organized infectious disease conference we organized in Basel, and he gave a talk about that paper. And what they did is they tried to better separate the different phenotypes of sepsis. And what they did is they took a huge amount of patients from various trials, published trials. So they had at the end 87,000 patients with suspected infection, and about 20,000 met the sepsis 3 criteria. And then they formed 4 new phenotypes, and I will explain them a bit in detail on the next slide. There's an alpha, beta, gamma, and delta phenotype. And these are characterized by specific laboratory or like physiological assets. So alpha phenotype, as an example, had fewer abnormal lab values and less organ dysfunction. So you can already see if there are less lab values and less organ dysfunction, the survival of the alpha phenotype was most likely better. And then the delta phenotype, here you had elevated serum lactate, transaminase levels. So clearly a patient who already may have lower pH, you know, a severely sick person also with hypertension. And so they came up with these phenotypes by doing something quite interestingly, and this is shown here. They took 29 variables from these published patients, demographics, vital signs, inflammation, organ dysfunction, and many more values. And then they looked into natural separation of these. So they had a statistical method where they could separate them, and really the alpha and beta phenotype, which you can see here on the graph on the very left side, is considerably, significantly different for certain lab values. Obviously, there are also factors where they are similar, but they try to determine the different phenotypes. And that was a very, very nice paper, I think, also showcasing you know, what you can do with retrospectively collected data. And this is also something which we do in the SPHN network, which you will see in a minute. So the impact of sepsis is very profound. So first of all, it has a high morbidity and mortality in septic shock. It can reach up to 70%. Also, if you survive sepsis, these people often struggle. I mean, being on an ICU is a traumatic event for yourself. So there's a loss in quality of life for many of these patients. And then obviously the healthcare costs associated with sepsis are absolutely substantial. I mean, if you think about how much a normal hospital stay costs, but on an intensive care unit, this is obviously much, much higher. Early recognition is therefore absolutely critical. So to identify the causing pathogen and to also determine the antibiotic resistant profile. So what are the current diagnostic bottlenecks to identify the pathogen and to also determine the antibiotic resistance? And for this, you first need to understand what the current diagnostic workflow is in a lab. And what we start with is a specimen, a sample, so a blood or urine sample. And here, what you can see here is the culture focused way of identification. So next you do a culture. So you take some material, you streak it out on an agar plate. And nowadays, this is not done manually, but automated by robots. There's a fair bit of standardization, and actually also now not a topic of this talk, but you can also analyze these images in an automated way. So you can, for example, say if there's bacterial growth on a plate or not. So there are modern diagnostic systems can do that. And then the next step is to identify the species. And what we usually use in the lab is mass spectrometry. I will not explain you now how this works here, but it's a very fast way, multi-TOF technology, and it works from a single colony. So you need bacterial growth, a single colony, and then from that single colony, you can also determine the antimicrobial resistance profile. And there are various methods you can use, and that also separates a bit from lab to lab. And this is also, as you know, an important aspect to make data interoperable. So for example, everybody uses a multi-TOF, but not everybody uses the same AMR testing. So obviously we need to know a bit what is used and how can we compare these results versus each other. Then another way, which is more direct, is detection of genes, and we can also sequence these genes. So that's possible. There are panel PCRs, for example, which allow you to assess, let's say, from a sample up to 20, 30 different pathogens in a relatively short time period. So here you bypass basically the culture step and you directly determine the DNA content. So these are basically the two ways how you would assess a patient who has a sepsis. Exactly. Now, culture takes time, and this is some data here from Basel, which I share with you. You can see here on the x-axis the time, and you have different box plots. The lower box plot shows you the identification, and if you take a look at where the medium bar is, so it takes 27 hours in median to determine a pathogen from sample collection until you know what the patient has, 27 hours. And if you remember now back the initial graph I've shown you, 27 hours without any treatment, the patient is dead if you do not treat them. So obviously what physicians do is they give broad spectrum antibiotics to empirically cover a large set of potential pathogens. And this also has a cost. There's no such thing as a free lunch, as you know. So you have antibiotic resistance, potentially side effects, and so on, things you need to consider. And therefore, we do antibiotic resistance testing, which takes in median about 50 hours. And then from that, you can narrow down the antibiotic let's say, focus and have a more targeted therapy. The other problem, what we have is the main, let's say, cornerstone in diagnostic is blood culture. So you take a blood sample and you culture that, and then from that you want to know if there's bacterial growth. First of all, the initial concentration in a sepsis patient is very low. You only have per milliliter of blood between 10 to 100 bacterial cells. So you can imagine these bacterial cells are outnumbered by the host cells by you know, many folds. And often we do not find a pathogen in positive blood culture. So we need to culture other material, respiratory material, and so on. And the time frame here is similar. I mean, you also need some time to identify, grow the bacteria, identify them. So there's a clear bottleneck in the identification process. So what are the solutions? We look into different types of biomarkers, not only molecular biomarkers. So we want to identify sepsis at an earlier stage, predict mortality, maybe help to tailor the treatment. And there are various ways where you could use new biomarkers. Things which we daily use in routine diagnostics, like C-reactive protein, procalcitonin. This actually assesses the inflammatory response of the patient. So you know that there's something wrong. The patient has a pathogen. You not necessarily know which one, but I mean, there are also, you know, new tests developed in this regards. Host transcriptomics is also something which is quite interesting, and there are a couple of startup companies promoting this. So for example, Inflammatrix, a very interesting setup of single genes they have there. And multiple studies actually have shown that some of the biomarkers only have limited value. And so I think this was clearly then our idea that we need new biomarkers for sepsis diagnostics. This is an important requirement. And we also need novel analytical approaches because you know, the data we generate with sequencing, for example, or all these omics technologies in general, they're very complex results. So we also need different ways to analyze them in a fast way. And because clinicians often need yes or no answers, especially in an emergency situation, you cannot have, you know, a lot of gray zones because you need to follow, for example, a certain treatment algorithm and then make clear answers are important. So this brings me to the topic now of the potential of digital biomarkers. And here, as you all know, many things have happened in the last couple of years. So big data is an important buzzword here. the volume of data we generate, the variety of the variety of data, the velocity how we generate data is remarkable. I mean, also in the diagnostics and in the hospital, also in infectious diseases, it's remarkable. And this was very, very nicely summarized by Eric Topol in a Nature Medicine review article, which I can highly recommend to read. I mean, he basically showcases that the diversity of data we have, and basically the benefit of data-driven analytical approaches to really come up with personalized solutions for patients. There's a huge issue with this, the explosion of knowledge in microbiology, and explosion of knowledge is a term which you often find in the literature. And we recently summarized that in a review article, and what that means for digital microbiology in a review article in CMI published this year. I wrote that together with Jacques Schrenzel and Gilles de Groip from Lausanne and Geneva. So this is basically the setup, what we can do. So there's a lot of potential and hope also in these biomarkers. And there are very nice examples. And I want to show you one example here, a study published in Nature Medicine 2 years ago. And that's, I think, an important and interesting study, which is retrospective. So it's not a prospective study. It's still therefore in a very controlled environment. And what the authors here did is they used the reinforcement learning for decision-making of sepsis treatment. So these were ICU patients. And the idea was that the algorithm learns how much volume, how much fluid a patient needs in shock. That's an important, important treatment aspect to provide fluids. But at the same time also how much vasopressors a patient need. And they trained the algorithm with, and you can see this here on the left side, they had a public available data set from MIMIC-3, and they included here about 17,000 ICU admissions. They had various models they validated and evaluated during that way, and at the end they came up with a model which worked. And verified that with 79,000 ICU admissions. So large public available data sets are used here, and this is also a motivation for us. I really hope that at a certain point SPHN will be able to also publicly share certain data, because then other researchers can pick that up. And so they included here in this decision tree 48 variables. And trained this algorithm, and it worked very nice. The AI was, in the selection of the treatment, was in average better than the human. But this is a retrospective study, and we have to be careful here, and certainly prospectively validate this. And the last author here, Aldo Faisal, also gave a talk to our sepsis network a couple of months ago, a fantastic talk where he also showed where this endeavor is going. And it's really impressive how these algorithms can also help and support clinical decision making in the near future. So here obviously an algorithm is used to support decision making, and based obviously on digital data patterns learned by the algorithm. So I think what is very important in digital biomarkers is to realize that we generate a lot of data which we reuse. So the vast amount of data routinely produced and collected can be reused and also reanalyzed with novel analytical approaches, artificial intelligence. The digital biomarkers may increase the efficacy of the diagnostic and therapeutic process. I've shown you an example now. I think it really allows us to recognize disease maybe earlier and also help us to treat the disease. And something which is very, very important by making reuse of routine data, this also is real world data. I mean, the physician has no other data, maybe, sure, you may have other data available if you examine the patient and so on, but having 48 variables is already quite complex. So I think it really represents to a certain degree real world data. And so what is needed to do that is infrastructure. So we need clinical data warehouses. we need data, and the data should be of high quality, interoperable, so curation is important, and we need experts. So data scientists will, I'm pretty convinced about that. In about 5 to 10 years, we will have data scientists in the hospitals, not at Google and Amazon maybe as well, but also in the hospitals. I think there will be new opportunities for data scientists, and who can actually, and then also we need people who can bridge, because talking two different languages, I think this is something which I realized during this SPHN project, how important it is to understand both sides and what are the requirements and questions we have. So this was the motivation actually to build up our project. Now I see here a question, I'm not sure, should I already answer the question here? Maybe we do the questions afterwards at the end of the talk. So the motivation of the sepsis project was to extract and integrate different data types of patients with presumed sepsis, which may then help to, first of all, better understand the complexity. I told you before that different age groups can be affected, and this is nicely showcased by these different phenotypes. We also want to develop multidimensional biomarkers. We'll talk a bit about that in a minute. To also think about which physiological compartments and time points are best for sampling. Because as I said, blood cultures only have a certain sensitivity in sepsis. And so maybe there are other compartments which are more important, and also the timing could be important for the sampling, because I assume that the bacteria in the blood are not constantly there. So there are perhaps times where you have more bacteria in the blood, to improve the outcome, obviously. So by earlier recognition, but also to predict who has a high risk for mortality. So you can change a bit the focus of care. Another motivation for us was to make the data interoperable between centers, but also between countries. And then set the framework for the future. So personalized diagnostics and therapeutic research. This really kind of summarizes the stuff I just told you as a motivation. Now, there are two parts, SPHN and PHRT, and let me quickly tell you what the specific aims are. SPHN has a very strong focus on infrastructure development, and this is also what we're going to build, or what we have built, is an infrastructure framework in the context of personalized medicine to connect the Swiss university hospitals. And here we have a very specific focus on intensive care units, because intensive care units are usually where severe sepsis cases end up in the hospital. So they are there to have a more focused care, in intensive care, and they are very well monitored. There's a lot of data produced on an intensive care unit. I mean, if you compare it to a normal ward, where you measure 3 times a day the blood pressure, for example, and in intensive care, you have a probe in your artery, and it constantly measures temperature and blood pressure. Then we also wanted to define ethical and governance standards. I will tell you also a bit more about our experiences here, and then complex clinical data sets, which will be harmonized to really have interoperable and comparable data between centers. And this is obviously ongoing work. It's not finished yet, but I'm gonna also update you on this. So there are 6 work packages, and the main point is really to collect data from the intensive care units, from the lab, from clinical diagnostic systems, to bring this into a data warehouse. And here I really have to thank again all the IT people doing this, and the IT experts in the clinical data warehouses, the data managers we had doing this work. This is very, very important to obviously store the data in a way, and it's not easy, in many ways not easy. And I think this is really improving, and it's fantastic to see how the progress is done here. So we also thought about, what data we want to collect, and this was the requirement analysis. Then we built this target architecture. Many hospitals obviously had an idea of data warehouses, but this was I really think also supported clearly by SPHN funding, and I think this is important to also realize that SPHN here has an important role also for the individual hospitals independent of the network. And then to connect, to build this connection. Now, PHRT has a bit of a different approach, and this is ETH money. And here, what they wanted to do is look a bit more into the scientific aspect, let's say. So there's a sepsis study, really like the clinical study, where we aim to recognize sepsis earlier, to predict mortality, to find novel biomarkers. And we have classical digital biomarkers I just talked about. We also have molecular biomarkers, which we will hopefully soon start with a clinical study, and then also hybrid biomarkers. Then selection for patients with, as I mentioned, like time points, compartments, etc. And an important part of the PHRT, and this I also think is very nice, that there's a feedback going back regarding the data quality. So how is this set up here? Obviously, the access of the data from, not directly from the clinical data warehouses in the hospitals, but actually the data is transferred to Leonhard Met, and from there it can be accessed. We have the digital biomarker discovery, we have molecular biomarker discovery, and here we have different groups involved, diagnostic groups, ETH groups, doing and performing different omics technologies. I will not spend too much time here because this is not work we have done yet, but obviously there's a strong interest in some of these technologies also in routine diagnostics. And then the reproducibility I just mentioned to you. So how is now our framework and setup? You basically see here the setup again, the SPHN part, strongly focusing on the infrastructure, bringing data from the primary systems to the data warehouse. From there, then the exchange basically of data. And here, just a few words. I mean, there are certain data standards defined, RDF, for example, but also the data is encrypted while being transferred. We have made some thought about de-identification of the data. So we do not report, for example, the date of birth, but just an age. Then there's also a shift in, so for example, if you collect data, today is the 29th of September, I think. So if you actually collect the data today, you would add in a change, so to make it more difficult to identify an individual by having these de-identification processes, and they are in place and working. And then the ETH component, the PHRT part, as I mentioned, and with omics technologies, with analysis of the data. And so this is really a nice setup with all the centers here actively included. Now, setting up a network is, such a network is, you know, quite a big project, obviously. So we had inputs from various sites. And what we did is, just to say, we had monthly telephone conferences, several face-to-face meetings, subgroups which were formed. And there was really a lot of discussion and also working in these areas. So clinical inputs, for example, and these are just, you know, a few examples here. It's not an extensive list or a complete list. We had what variables are interesting to be collected. You saw before, most studies use between 40 and 50 variables. What are the research questions we have? Then technical inputs are, how do we organize the variables in a data model? How do we transfer the data in a safe way? Then the analytical inputs, what methods can be used to analyze the data? What data quality aspects can be improved? So there are various questions which we then try to answer in discussions. So let me talk a bit about the current achievements, what we've done. I think if I start now with the ethical and legal framework, I think the legal framework I personally have underestimated. It was really a difficulty with the different legal services to find, you know, to actually define a contract. The ethical committee of Switzerland, when they assess such a project, they always want a consortium and collaboration agreement between the centers and a data transfer and use agreement. And in such a large network, obviously there are many players involved, and people also think, what can you do with the data, etc. And it really took us some time. I think this is a major achievement that we have set up here, a contract between these centers, and it delayed the project, yes, but I think now we are absolutely on track, and this is pretty good. The digital biomarker study here in the ethical framework, this is regulated by Article 34. So this is a data access is possible because this is routinely collected data. And the omics biomarker study, there we will collect additional blood sample, and this will be consent-based. So we will ask patients, not necessarily when they enter the intensive care unit, because that's hard to do, because often they cannot provide the consent. But we will actually ask them later if they survived sepsis, and if they did not survive, we will ask the relatives if they agree that the samples collected and the data can be used. So we developed these documents which are listed here, the consortium agreement. We also defined the scientific and executive board. We had the data transfer and use agreement. We have a publication policy. This is also very helpful, and the digital biomarker study is fully approved. I'm going to show you there a bit more in terms of what data we have collected so far. And the omics study is partially approved, and in Basel, for example, we are setting up now a test trial to include a patient and to really go through the workflow. So the data focus on sepsis, so there's regular data release from the IT teams with a focus on infectious disease and sepsis. So the idea was to have a shared concept between all the centers, to also include new concepts which go a bit more into clinical details. So for example, antibiotic drug resistance is also a very important topic in sepsis management, and these are specifically developed data concepts for a sepsis network. And also what is something which we are continuously working on is the interoperable and interconnected data set. And then the transfer via the Biomed IT nodes, as I mentioned, to Leonhard Met with de-identification, with encryption, which is also ongoing. Now, how many patients have we included? And I'm really proud of that. I think it's fantastic. In November 2019, we started, and we have around 6,000 patients are included. And this is a massive data set with a very rich time series data of patients on the ICU. Just as an example, I think, if I remember correctly, from Geneva, there were about 700 patients, and this includes over 2 million oxygen measurements. So this is a very, very rich data set, and it will be fantastic to analyze and to learn from it. And the cool thing about this data set is obviously there are not only patients with sepsis, because we do not know who has a sepsis in the beginning. It also includes patients who do not have a sepsis. So it's everybody on an ICU. So patients with a sepsis are the cases, and patients without sepsis are a control. And because we have this data, this is also an interesting data set to explore other critical care diseases, let's say stroke or myocardial infarction, or if you have a car accident, let's say. These are interesting data sets which can be analyzed. So how to change the workflow in an ICU, that's also an important part. So one thing is, I mentioned that to you, that we need to have a suspected or a proven infection. So what we try to establish is a sepsis tag on the ward, which is on the ward, so that you actually say this patient has a suspected infection. But on the other hand, you could also use potentially certain surrogate markers like blood cultures, which are collected, antibiotics, which is administered. And it will be very interesting to compare these two and see, could a surrogate marker also, perform a similar here to identify certain patients. And then quality control over time is also very important. So for example, is the data, can we identify it? How well is it structured in the different centers? And this feedback will also be important now when we go and start analyzing all these rich data sets. So data collected so far, here I just said, I mean, the overall number is around 6,000. We have demographics, lots of information on sepsis criteria, drugs, laboratory data, vital parameters, and these are organized in sort of data releases. We are still waiting for the Zurich data. This is in preparation, but all the other centers have shared this information. And whenever we have a new data release, obviously, you know, the patients already included, this data will also then be added. So it's not that we then have missing data of these 6,000 patients. Let's say if the next data release would focus on antibiotic resistance, let's say, all the 6,000 will then have this information available. And there are specific data concepts developed from SPHN. There's a data concept, but obviously also PSSS, the sepsis project, has a series of complementary data concepts which are very important for infectious diseases and can also be used in the future for other infectious diseases. Maybe somebody comes up with, I don't know, a SARS project or something else, then these data concepts can be very much of value. Then we have, in the beginning, determined a huge amount of variables which we are interested in. We identified more than 500 different variables which were defined, and obviously we are continuously working on it, and I'm pleased to say that a large part has been uploaded and generated in this, let's say, well, last couple of months. The data managers here really also focused on this list, which was defined by the clinics, and I'm very thankful that this worked so well. So what are the summary and conclusion here of this project? Quickly check the time here. We obviously try to form a Swiss-wide network for sepsis research, and the driver project here generates a unique framework between multiple disciplines and institutions. This is fantastic. We have a strong focus on clinical routine data and infrastructure development by SPHN. We have a strong focus on omics data by PHRT. Here I have to say, not every patient, or not of all 6,000, we have this omics data. So this is really a clinical trial with about 400 patients where we will have this omics data available. But nevertheless, I mean, this will be a super valuable and rich data set as well, where we can maybe use then future biomarkers from. So what are the next steps? We will improve the data interoperability. We will also start now to analyze the digital biomarker discovery phase and also start the omics study. So I think these are the next immediate steps for our network. What is critical for SPHN and PHRT in the future? I think what is very important is that we use other primary data sets. So for example, in a hospital, I mean, the patient ends up in the ICU in the worst case, but obviously the earliest division seeing a patient is the emergency department. So, and many emergency departments still have, you know, paper sheets, etc., but they are more and more digitalized as well. There are monitors in an emergency room. And if we can integrate these information into the data sets, that would be fantastic, but that's maybe something for the next SPHN funding period, because, you know, obviously we had funding for the ICU. Then smaller and mid-size hospitals, that would also be interesting. So for example, cantonal hospitals, there are also very large cantonal hospitals, obviously, which could be very interesting to include here. And then I also think something which may be interesting is, you know, we are always in these data-driven project focus on the disease state of a patient. So we have a patient who is severely sick, but obviously he was healthy before, and maybe there are ways to detect something is not okay in very different ways. So for example, smartwatches, which can detect heartbeat differences. Perhaps this could be used to also have an early warning for sepsis. So this whole aspect of citizen health and citizen science comes here into play. Then I think the SPHN family should be merged with the networks. I mean, yes, I know this, the data set is set up this way, but there are fantastic oncology data sets. And obviously, some oncology patients also will develop sepsis, and maybe the other way around. A sepsis patient may have an oncological problem. If you have, for example, E. coli in your blood, this could be an indication that you have a urothelial cancer, for example. So this is quite interesting to actually see here also down the road, the merging of the data sets. And then funding, until end of 2021, we have funding, but what then? So clearly we will need more funding to solidify the SPHN network. I think sustainability now here is key. Maintenance and curation of data is costly. So there is obviously a competition in healthcare. I mean, the different university hospitals not naturally work together. So why should they work together? And I think we need to generate a business case for each hospital. It sounds a bit tragic, but business cases are important in healthcare. So they should benefit from the own data they collect in the clinical data warehouse. And I can say that during the SARS crisis now, we very much benefited from the clinical data warehouse in many ways. So they should also benefit from data sharing to realize that they can actually tackle problems by sharing data. And what could be incentives for data sharing, maybe on a technological problem. So it could be that there are general aspects of digitalization which a hospital tries to improve, and with such a network that could they could actually have here a boost in their digitalization. Blockchain technologies, I mean, that offers tokens, so perhaps there could be some interesting ways by sharing data that they get more tokens, for example. And then on a scientific level, obviously improve patient care, reduce costs. If you can reduce the amount of patients going to an intensive care unit, you will drastically reduce your costs in a hospital, and that could be a motivation. And then, and this is maybe very controversial, but why not commercialize some of the data? So really selling patient data to companies, I mean, or develop products together where you could also maintain such a system. I mean, I'm not saying that we will, anonymously, obviously, and with a lot of, proper framework, etc. But this could indeed be something which could be interesting to maintain and keep long-term funding available for such a study. Funding agencies, I think at the moment there are many cohorts available. If you think about the HIV cohort or the transplant cohorts, perhaps there should be a funding slot from SNF on digital cohorts. I don't know, database funding. Maybe this is something SNF should really think about, especially now that these data set, the maintenance of such data set is costly. And then I think really our framework needs another round of seed funding to solidify the good work we have done. I think it would be fantastic. I think we made huge progress, but at the end, with another couple of years, this will become a very strong foundation for many, many things here. So I think it's now important to acknowledge a lot of people. I listed here people from my research group, which are at the University Hospital of Basel, and that have a lot of exchange with them in bioinformatic questions and so on. In Basel, obviously, Professor Batke, Hans Paco, Martin Siegemund, Bram Stieltjes, and this whole team. I also want to especially mention Rita Achermann, who is a very important person for me in the interaction with all these SPHN related questions. And it's really fantastic to work with them all. And then the SPHN network, all the intensive care units, infectious disease people, clinical microbiologists. I just here listed the clinical people, being aware that there are a lot of people in the background also working. And I also want to thank the SIB and DCC and ETH Zurich and all the other involved teams, which I have not mentioned here. So a big thanks to them. And if you want to follow me on Twitter, I think I have like 50 followers. Please follow me. And then I will also regularly update here a bit more in the future on SPHN achievements. And thank you very much for attending. And I'm happy to take now, hopefully, a lot of questions.